The first of the two lectures is on new foes, and then the second one will be on the progress we've made towards eradication. So again, just to lay out the lectures, this is number 3, and then there will be number 4 afterwards as well. And Charles Penn is doing number 5 tomorrow. It's annoying, but the tomorrow slot at 1 o'clock clashes with our seminar series. So it actually is a very good seminar that you should have been able to go to, but unfortunately, because it clashes, I think Charles looked into rescheduling, but he hasn't been able to do that. So he's disappointed, and you should be disappointed, but there we are. And because I'm chairing that meeting, I have to go to that. So, what do we mean by emerging infectious diseases? Well, there are various definitions or various ways in which infections fall within this umbrella term. There may be diseases caused by newly identified species or strains. Some listed there: SARS, AIDS, Ebola, Nipah, E. coli 104. Or there may be a variant form of an existing organism that suddenly takes off. So we might get pandemic influenza because we have a new form of influenza where there's been shuffling of genes to give us a new configuration. It may be that we have an infection that we already know all about, but it moves to a completely new geographical area. West Nile virus is an example of that, where this has spread through North America over the last couple of decades and moved into completely new areas. It may be that we get re-emergence of infections that we knew about in the past, but where they've been quiescent recently, either because of emerging drug resistance, so multi-drug resistant tuberculosis, or because there's a breakdown in public health. So in Haiti, for example, we've seen cholera at levels which we've not really seen for many a year anywhere else because of breakdowns in public health. And sadly, that's supposed to be likely to get worse with the hurricane damage that's there. There's a paper there which I've given you a link to if you want to look into this in more detail. What kind of factors make diseases emerge? Well, one clear issue is us as humans and what we're doing and how we're moving about and how our populations are changing in their structure and where they live and so forth. So if you go back, I don't know, say 100 years or 150 years, kind of the average person in England might move from his village to the next village, maybe go to the nearest town occasionally, but that would be it. And they would probably do that 150 years ago. They'd have done that with a horse and a cart, or they'd have walked. 100 years ago, they might have started going around in an automobile and might have gone and occasionally gone a few hundred miles, gone down to the Big Smoke in London or whatever. But for the last 50 years or so, people like us have been jumping on aeroplanes, and you can get to the other side of the world within 24 hours. And so it's clear that those kind of changes in behaviour are going to have an effect here on how diseases can spread. There are obviously changes in our susceptibility to infections. AIDS, HIV infection renders us immunocompromised, which means that people will start getting infections that they wouldn't normally get if they had normal immune function. Similarly, in a very constrained context, the context of patients in hospitals who are receiving treatment for cancer, cytotoxic agents, that will make them immunocompromised. And again, they then become susceptible to pathogens that we wouldn't have recognised as pathogens before. Climate change is a big hot potato at the moment in terms of how much effect that's going to have on patterns of disease. If mosquitoes can start overwintering in places where they couldn't before and so forth, all kinds of changes. Economic development and yet land use. So the fact that humans are starting to encroach on the wilderness as our populations grow and as our cities expand, and that some of you may have seen, I think it was it this weekend or the last weekend before, about we might start seeing wild boar coming into British towns and cities because they're getting adapted to us and we're getting adapted to them. Breakdown of public health, that's the sort of thing we've seen in Haiti. Abnormal natural occurrences, earthquakes and volcano eruptions and tsunamis and hurricanes. And then, of course, what humans do deliberately. So war and the devastation caused by war and the release of organisms into the environment through bioterrorism. Thankfully, at the moment, that has been a very minor player in infectious disease. There's only been a handful of cases like that. But Martin Rees, who was used to be president of the Royal Society, has a kind of bet that sometime in the next 100 years there will be 1 million deaths from bioterror or bioerror. So we can't discount it. Okay, I'm now going to have a bit of an interlude and show you a film. Except that the film doesn't want to work. Well, come on.
It was a groundbreaking ceremony for a new factory.
Did you mention seeing anyone who was sick? Anyone on a plane, the airport?
No, she said she was jet-lagged.
The average person touches their face 3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, and each other.
Back. No, go up to your room, honey. So we have a virus with no treatment protocol and no vaccine at this time.
You had a seizure this morning, Beth.
Does she have a history of seizures?
No.
Allergies?
As of last night, there were 32 cases.
Unfortunately, she did die.
Right. Can I go talk to her?
Mr. Amoth, your wife is dead.
What are you talking about? What happened to her? What happened to her? Is there any way someone could weaponize the bird flu? Is that what we're looking at? Someone doesn't have to weaponize the bird flu. The birds are doing that.
Watch this. It's transmission, so we just need to know which direction.
On day 1, there were 2 people, and then 4, and then 16. In 3 months, it's 1 billion. That's where we're headed.
They're calling out the National Guard. They're moving the president underground. People will panic.
Get away!
It will tip over.
The truth is being kept from the world.
Cook your samples, destroy everything.
Hello. I need you to get me the names of everyone who serviced this room. It's an emergency.
You can't panic now.
I know.
I'm going to get you home. I got people too, Dr. Cheever. We all do.
Don't talk to anyone. Don't touch anyone. Stay away from other people.
Get back in your car. We're not sick!
It's figuring us out faster than we're figuring it out.
It's mutating.
And look, that film was actually based on 2 different kinds of disease. So the symptomatology belongs to a disease called Nipah, which I'm not going to say much about. It's a disease that surfaced in Malaysia, associated with pigs and bats. But the transmission was much more based on what we saw with SARS, which has been called the first pandemic of the new millennium, and focused on this place down here, Guangzhou. So in November 2002, the first cases of this particular disease were seen in southern China. The Chinese authorities were rather slow in reporting problems. They preferred to just keep things quiet. And in fact, the People's Republic did actually apologise to the world for not actually looking at this in depth quickly enough and spreading information quickly enough. By February of 2003, there had been an outbreak in Guangzhou hospitals involving patients and healthcare workers. And by then there had been a cumulative number of cases, over 300, a third of which were in healthcare workers, and 5 of them were dead. This was from an unknown acute respiratory syndrome, and it wasn't clear what was going on. It didn't look like anything that people had seen before. It looked like it was transmissible. Now, if it had been stuck in China, that would have been bad, but things got a lot worse when it decided to start jumping on aeroplanes and travelling around the world, this particular infection. So on the 15th of February, a 65-year-old professor of nephrology from Guangdong province started to fall unwell. I think, if I remember rightly, he was going to a wedding of his nephew in Hong Kong. He resided at this hotel here, sometimes called Hotel M. It's in fact the Metropole Hotel in Kowloon in Hong Kong. And he was there for just one day. And by the second day, the 22nd of February, he had gotten so ill that he couldn't go to the wedding. He decided to just take himself straight to hospital. But in those that less than 24 hours stay, he managed to infect 17 residents at the hotel. The contacts of that individual at Hotel M then went on to travel to Hanoi, Singapore, and Toronto. And in each place, they're starting new outbreaks. So one of the Hotel M contacts, a US businessman called Johnny Chen, fell ill on a flight to Singapore, and he was actually admitted to a hospital in Hanoi and died there. Another contact went to Hong Kong and started an outbreak there in the hospital. Another went to Toronto and died there. And 5 members of the family were affected. So within a very short period of time, this agent had spread to 3 more international cities. Perhaps one of the most poignant stories within this outbreak was the story of this guy here, Carlo Urbani, who was an Italian doctor who worked for the WHO in Hanoi. And he had seen there an explosive nosocomial outbreak. Nosocomial is just a fancy way of saying hospital outbreak in Hanoi. And it was his description of these cases outside of Guangdong that actually kind of rang the alarm bells across the world. That was basically what led to the acceleration of research to identify the virus, combat the disease. So his work there actually probably saved thousands of lives by actually bringing this to the world's attention so quickly. The sad thing was, though, that he actually succumbed himself a month after seeing his first case. He went off to a conference in Bangkok and fell ill on the plane and died there. A little while before that, he'd actually said that, people said, you're very brave and doing all this. And he said, if I cannot work in such situations, what am I doing here? It's a job just answering emails and going to parties and pushing paper. So in a sense, he was one of the heroes of this. And his intervention actually made a decisive impact. A few weeks later then, WHO issues a global alert, and the name Sudden Acute Respiratory Syndrome is coined to describe this new syndrome. It's a bit of a nondescript title, and I guess it's lucky for the people of Guangdong and Guangzhou and Hong Kong and whatever that it didn't get given a geographical name, which is quite commonly the case, and then all sorts of political ramifications come about if it being called Guangdong fever or something. Anyway, there was a call for a global international research effort, and scientists working in Hong Kong, at the University of Hong Kong, were the first to isolate a new virus from an open lung biopsy. This was unexpectedly belonged to a known group called the coronaviruses, which up until that time had really been seen as rather minor players in human disease associated with cold-like symptoms. This was soon confirmed in the US and in Germany, and the genome sequence showed that this was distinct from all known human pathogens. So within a few more weeks, they had a full genome sequence. It turned out that a virus almost identical to that in terms of genome and so forth was then identified in an animal reservoir, the palm civet, and various other game food animals. That provides some context. It's said that people in southern China, in Guangdong province, will eat almost anything. If it flies, they'll eat it. If it swims, they'll eat it. If it crawls, they'll eat it. And there is this, particularly with the economic growth in China, there is an interest in eating exotic things to show how much money and influence you have that you can get these things. So the palm civet had been on the diet there, and that was fingered as a potential source. And in fact, they were slaughtered. They were stopped from being being sold, and then there were massive kills of these were being farmed, and so all the farmers had to go and kill their stock. By July of 2003, there was a lack of transmission in Taiwan, where the last cases were sputtering on. That was the end of human-to-human transmission, so that was the end of this SARS outbreak across the world. Unfortunately, there are risks in trying to do research, and in Singapore in 2003, September, there was escape from the lab. Towards the end of 2003, beginning of 2004, there were some new cases in Guangzhou from animal to human transmission. Another lab-acquired case in Taiwan, and then the following year, some in Beijing as well. And then finally, in 2005, it became clear that actually the ultimate source was not the palm civet. The palm civet might have been a kind of amplifier organism that got the infection somewhere else, but that bats, and particularly horseshoe bats, were likely to be the actual environmental reservoir for this particular virus. So that's just a summary here of the whole global picture. And you can see that there were cases across 3 continents, 4 continents, in fact, there, North America, South America, Europe, Asia. There was local transmission. So it's spread from patient to patient in Toronto, Ottawa, San Francisco, Ulaanbaatar in Mongolia, Manila, Singapore, Taiwan, Hanoi, and Hong Kong. And within the People's Republic of China, they were spread to multiple provinces, Guangdong, Jilin, Hebei, well, I won't read them all out, but they're all there. So what about the virus that caused this? The SARS coronavirus. Coronavirus is thought to be benign. This is a large enveloped positive-stranded, single-stranded RNA virus with a relatively small genome, encoding a replicase and some structural proteins. And it was discovered that the S protein, the spike protein, actually binds to a particular receptor on host proteins, ACE2 receptor. And from sequencing the genomes of viruses isolated during the epidemic as it progressed, it was found that there was actually evolution going on. So mutations in that S protein were showing that the organisms were becoming more adapted to being transmitted between humans as the outbreak continued. And looking at neutral changes, things that weren't to do with actually making the virus more fit in its environment, those actually allowed scientists to reconstruct the phylogeny of the outbreak and actually trace its spread. So what about the disease? Well, the disease was defined symptomatically to begin with, because that's all they had to go on. There's a case definition put forward. Incubation period of 2 to 14 days. Flu-like symptoms, a high fever, myalgia, which just means muscle aching, lethargy. Gastrointestinal symptoms, a cough and a sore throat, shortness of breath. Tied in with that, there has to be, within the last 10 days or so, close contact with a probable or suspected patient, or been in an area where the disease has been transmitted. So these are kind of very much ad hoc definitions, but they kind of work during the outbreak. And most of the patients had an abnormal chest X-ray. And then within a few months of the outbreak, once you had all those genome sequences and isolation of the virus, you could then start to have laboratory-confirmed SARS. How does it get transmitted? Well, human-to-human spread. There's direct or indirect contact of the mucosa with infectious droplets or fomites. Fomites is a fancy epidemiological term for kind of everyday objects that can pass from one human to another. So, books, clothes, household objects, that sort of stuff. It seems to survive quite a long time in the environment, much longer than other human coronaviruses, so 2 to 3 days on dry surfaces, 2 to 4 days in stools. In Hong Kong, they focused their attention on a particularly explosive outbreak in a particular housing estate called the Amoy Garden housing estate, where there were hundreds of cases, and it appeared what was happening here were there were these dried out U-tubes, U-traps in the sewage drains that were supposed to obviously stop smells coming up from the drains, but they dried out completely. And then there were exhaust fans in the toilets that were generating aerosols, and the aerosols were going up a light well within the building, so that they were sort of being wafted out and then wafting up this light well. So it was getting onto different floors of the building. 5 episodes of SARS actually being transmitted on commercial aircraft as well. So again, it seems to become battle ready for being a globetrotter. So the probable origin is a cross-species jump from civets, probably as the proximal host, and then as the ultimate host, horseshoe bats to humans. The second case was a chef with multiple animal contacts. And here you can see a phylogeny of based on sequences from various different coronaviruses. And you can see here human SARS, civet SARS, and bat SARS all very closely clustered together. And then a few other bat viruses as well. So how was SARS controlled? Well, the principle was to break the chain of transmission. And they had a 3-step protocol: case definition and detection, prompt isolation, and contact tracing. Unfortunately, there wasn't much in the way of treatment. Various treatments were tried. It's unclear how effective they were. Interferon was used, steroids, ribavirin with protease inhibitors, and convalescent plasma containing neutralizing antibody. I haven't actually managed to scratch beneath the surface to find out whether there was any good evidence that any of those worked. These are sort of rational things to try, but whether they worked or not is unclear. In terms of epidemic containment, in each area there was an emergency operating centre for control of the thing with institutional support, and there was effective quarantine measures. So in Hong Kong, Canada, Singapore, Taiwan, thousands of people were quarantined in their own homes. In Hong Kong and Singapore, they closed all the schools, and they brought in legislation very quickly to actually control the outbreak. The WHO also played a role in international collaboration. There was travel alerts and restrictions. Controversially, the WHO actually advised only essential visits to Toronto from the outside world. So effectively the whole city of Toronto was placed in quarantine. And airline passengers were screened for fever using thermal imaging scans and taken to one side if they happened to have a fever. There was coordination of research as well, and agreement between countries on how to actually contain this. So the aftermath, where did it end up? Well, we ended up with an epidemic that had affected 37 countries, over 8,000 cases, nearly 800 deaths. So that makes a case fatality rate of about 10%, which is pretty appreciable. There aren't many diseases that we see transmitting around that have that kind of level and go this global so quickly. Why did it happen? Well, there is an ecological and an economic reason, if you like, which was that the rapid growth in China, economic growth there, primed this demand for exotic food. Well, for food in general, for meat in general, but particularly for exotic foods. The way in which those animals were housed in markets in overcrowded cages with no biosecurity meant that it would be very easy for one civet to infect the next one. And obviously, the fact that the virus could jump from animals to humans made this possible. And that's a sort of, that's a contingent thing. You can't predict which ones are going to do it, but this one could obviously do it. The rapid global dissemination depended on the fact that it could transmit from humans to humans very quickly. There was some deficits in infection control in hospitals early on, I think. But of course, international air travel was a major component of all this. And so those kind of things, the film Contagion there, it's spot on in the fact that, if we did get one of these kind of things going on and people jumped on aeroplanes, it would be all over the world very quickly. And this really did have an unparalleled dramatic impact on healthcare systems, economies and societies of the affected countries. So are we ready for it next time? Could it come back? Well, if the conditions are right, then it could. The animal reservoir is certainly I mean, whether it's still out there in wild civets is unclear. It's probably the domesticated civets that were the proximate host here, but in wild bats, there's no reason to believe it's gone, and we can't sterilise the world of bats. Next time we will have the wherewithal to diagnose and respond much more quickly. In fact, there have been over 4,000 publications on SARS since the outbreak began. So in terms of the knowledge to deal with it, we have that. But there are still gaps in our knowledge in understanding how it's transmitted, how it causes disease, and screening tests, infection control. We don't have a safe, effective vaccine. And we don't know really where it's living out there in southern China and how likely that is to get back into to humans. Now, I'm going to take a break now because this is 2 hours, and I'm going to get a friend of mine, Jennifer Gardy from University of Vancouver, University of British Columbia in Vancouver, to tell you about swine flu in 2009. Now, is this going to work?
They were doing
it is
all
going
right.
to work. That's good then.
So Richard's talk really brought up two things. It brought up that there's a lot of new technology that's out there, and there's a lot of new attitudes towards technology, and that public health in the 21st century really has to capitalize on this new technology and on these new attitudes to become basically public health 2.0, a new model for delivering public health services. So in listening to Richard's presentation, I was reminded of something very similar to this thought of this new model of delivery that I had written about H1N1 back in May. And so in my other life, when I'm not doing CDC-related things, I blog for the Globe and Mail's globecampus.ca website as Nerd Girl. It's a lovely photo of me with very good lighting. I don't normally look that good. And back in May, when swine flu first broke and appeared on people's radar, I wrote a post about some of the things that, as a scientist, I found really fascinating about the current outbreak. And one of those things was the idea that this H1N1 outbreak is really world's first open source outbreak. And what I meant by that was that data about this virus was being generated in real time, and it was being shared with researchers around the world freely with no restrictions, and the community had really come together as kind of a hive mind to solve this problem of pandemic influenza. So reminded of this idea that I had by Richard's talk, I figured that this would be my suitably TED-worthy topic. So what I'm going to talk about today is this idea of the open source outbreak, or outbreak 2.0. What is it? What are the factors that have enabled it? And how did it really unfold with H1N1? And what might we see as far as outbreak 2.0 for the next outbreak? So I figure I'll give you my take-home point right now, no sense in waiting till the end, and that is basically that advances in technology, particularly around genome sequencing and just bioinformatics and infrastructure, have really contributed to this, as has a sea change in scientists' attitudes towards becoming more open access oriented and more collaborative. And really those two things together have brought us to the brink of this new era in public health, where our response to emerging infectious diseases and emerging threats is much more rapid than it's ever been in the past and much more collaborative than it's ever been. So we'll hop in our awesome time machine and go back with Doc to the heady, carefree days of March 2009, when swine flu was at nary a blip on anybody's radar. I've got a calendar here. There's March, April, May, June, and mid-March that public health officials in Mexico started picking up increased flu activity, and this increased throughout late March and early April. And on April 15th, the first two cases of what would ultimately be recognized as H1N1 were picked up in California, and the screening program in California kind of triggered a warning bell, because these were two cases that didn't resemble any previous human influenza that we had seen before. So the Californian officials called in the CDC, and about 6 days later, the CDC was able to confirm that this was a novel swine origin influenza virus. So as soon as they issued this advisory, public health officials, basically the world over, started looking for this virus in their own population, and sure enough, they found it. They found it here in Canada, they found it elsewhere in the States, they found it in Europe, and by the time the 29th rolled around, just over a week later, it had basically been found everywhere. You know, over that week, the number of cases was pretty much doubling every day. So on the 29th, the World Health Organization raised the pandemic threat level to 5, meaning that a pandemic was imminent. And as we all know, the number of cases steadily increased throughout April and May. And finally, on June 11th, a pandemic was officially declared by the World Health Organization. In Canada, as of June 11th, when the pandemic declared, at that point we had about 5,000 laboratory-confirmed cases of swine flu. And using a figure of 20 to 30 cases out there in the wild for every lab-confirmed case, that meant we were looking at about 100 to 150,000 people that were infected at the beginning of June. So where does the idea of open source outbreak fit in with all of this? Well, if we go... We'll go back to our calendar in a second. I love this shot of the sneeze. It's so disgusting. It's the perfect public health picture. As people were sharing germs and this pandemic was spreading around the world, so too were researchers sharing data. And if we go back to the calendar, we can see that on April 25th, just like 4 days after the CDC confirmed this novel infectious agent, we already had the first genome sequence for swine flu, the blueprint for making the swine flu virus. And the very next day, this is so cool, a group of international researchers, there's 13 people representing 8 institutions across 4 countries, came together and decided that they were going to take this genome sequence data and apply molecular evolutionary analysis techniques to it to figure out where this bug had come from and how it was evolving. So they created a wiki site. It was basically an open lab notebook that was open to everybody in the world. They could check out the analysis that this group was doing, and they worked 24/7 on analyzing the genome sequence data and figuring out what was going on with swine flu. As a matter of fact, that very same day, that group working together as a team had figured out the origins of H1N1 influenza. They realized that this virus was made up of 6 parts of a pig virus that had been circulating in North America for a while and 2 parts of a pig virus that had been circulating in Europe and Asia. Only 5 days after the first genome sequence was made available, we had the first paper, official science research paper, published on H1N1. This was, again, about the origins of the virus. It was a different group from the wiki group, but they arrived at the same conclusions. And this is absolutely remarkable, to go from a genome sequence and 5 days later having a peer-reviewed and published paper using that genome sequence. It's the fastest turnaround we've ever seen in science. By about a week and a half later, you had genomic data available for about 69 different viral isolates, and the wiki group used this to do a molecular clock analysis where they calculated that the virus would have entered the human population probably towards late 2008 or early 2009. And this molecular clock analysis was included in really the first major paper about H1N1 that was submitted to the journal Science on May 5th, and in only 6 days it had been peer-reviewed, accepted, and made available online. Again, remarkable turnaround. And a month after that, by the time the pandemic was declared on June 11th, you had over 250 papers available on H1N1. If we go back and compare this to SARS, which was our last big outbreak, you'll see just how quickly we've managed to improve our response times. So SARS, we thought we were pretty fast with SARS. If you take day 0 as the day that we isolated the SARS coronavirus, it took us 19 days to get to a viral genome. And I will give a gratuitous plug here. That viral genome was done at the BC Centre for Disease Control with researchers from the Genome Sciences Centre. In 19 days, yes, I can't take any credit. Applause to Bob. Good job, Bob. That's the reason we're all still here and not dead of respiratory illness. In 19 days of H1N1, we had over 100 viral genomes. We knew where and when and how this virus had emerged. There were multiple papers that had already been published or were in press, and we were already growing up a seed strain for use in the vaccine. So, we thought we were fast in 2003, and we're really fast now. So how is this possible? How are we so fast at responding to this? Really boils down to 2 things: technological advances and a shift in the attitude of the science community. Technology-wise, it's never been easier, cheaper, or faster to sequence a genome. Back when the Human Genome Project started in 1990, it took 10 years to get to a draft version of one human genome. It took another 3 years to complete it, though many would argue it's still not actually complete. Cost $3 billion and took hundreds, if not thousands, of people. Fast forward to spring of this year, it took this guy 4 weeks, $48,000, and 2 of his friends to sequence his own genome in its entirety. The advances in this field have been absolutely remarkable, and they just keep coming. I mean, it's getting to the point where probably within the next 2 years, we're going to have a $1,000 human genome available within a week. Similarly, it's never been easier, cheaper, or faster to push data around the world. We've got things like email, collaborative workspaces like Google Docs and Wiki, and we've just got the network infrastructure to support all this. I mean, when I first got on the internet, and I won't tell you when that was, it was a long time ago, I would hit the Get Messages button on my email program. I would go downstairs and make a sandwich, and then I would hope that by the time I'd finished making that sandwich and came back upstairs, my 6 email messages would have downloaded, and sometimes they hadn't. Similarly, along with these technological advances, we've had real advances in scientists' attitudes, and you're seeing the ranks of academia and research populated by people of my generation, and for lack of a better word, I'll call us the file-sharing generation. So we were people that grew up trading our Commodore 64 games back and forth with each other. Totally dating myself. I look young, but I'm older than you'd think. We trade our Commodore 64 games. We go on BBSs, and we were the first people to use Napster and create Friendster accounts and things like that. And this really engendered in our generation this belief that information, and particularly knowledge, wants to be free and should be free, and that there should be no barriers to accessing it. And this has really manifested itself in research as an open access, and really the adoption of an open access mentality. Another gratuitous plug is that it's Open Access Week. In 2 weeks, UBC's got a lot of excellent events planned, so do check them out. Back when I was starting graduate school, PLOS, the Public Library of Science, and Biomed Central, both open access publishers, these were just fledgling concepts. They didn't exist yet. But now, just really about a decade later, these have become 2 of the biggest publishing forces in science and publish some of the most impactful journals that are out there. And in fact, a survey that was done earlier this year says that 85% of scientists support open access. So in addition to the spirit of wanting information to be free, scientists these days are also really big on collaboration. We appreciate that, 2 heads are better than 1, but 20 heads are better than 2, especially when those 20 heads all come from different disciplines and bring different skill sets. And I think this is really clearly illustrated when you look at how authorship of papers has changed over the last 50 years. So I have on the left a screenshot of Nature taken from an issue published exactly 50 years ago today, and you can see that there's 2 articles on this page whose authorship you can see. They're both single author papers, and it's kind of hard to see the references, but they all refer to single or dual author papers. On the other hand, you've got a paper taken from Nature, the issue that came out on Thursday, and this is much more typical of what you're seeing these days, and this is a paper with 36 authors. So we've really embraced collaboration and team science. Science has moved away from the domain of the individual and moved into the domain of the community. So how is all this, genome sciences, technology, data infrastructure, and changes in attitudes impacted our response to H1N1? Thanks to all the data that's being generated and shared in real time across the world, we know that as of this morning, that the virus is still susceptible to all the antivirals that are out there. We know that it hasn't drifted from the vaccine strain. It hasn't evolved away from that, so that the vaccine will still offer protection when it's available. And we know that it's basically entirely displaced seasonal influenza, and it's the only flu that's circulating around. What's really interesting is that in any outbreak situation, it's always a race between the spread of information and the spread of the virus. And in the past, the virus has always won that race. It's always gone a lot faster than the info has. But this is the first time, really, where the spread of information has caught up to the spread of the virus. And I think in future outbreaks, you're actually going to see information travel faster than the bug can. So what about the next outbreak? We don't know what it's going to be, but how are we going to deal with it?
So Jennifer gave that talk in 2009. I actually met her last year at a meeting in New Orleans, and I was particularly taken by the way in which she described all that. I met her early in the year, and then by the summer of last year, we actually had our own experience that was very similar, and the way in which she was thinking about it actually primed our thoughts quite a lot. So let's just focus the last few minutes on another disease, infectious disease, in hemolytic uremic syndrome. So this is linked to a particular kind of E. coli called Shigatoxin-producing E. coli, or STEC. Now, STEC causes diarrhea. Typically, the diarrhea is a bloody diarrhea, and you also get the toxin, as well as causing local damage in the bowel, actually spreads through the body and causes damage to the kidneys and the brain, causes anemia because it leads to damage to red cells, and you get loss of platelets. So this is the 3D cartoon of the toxin. Now, last year, in May through to July, Germany was in a very unfortunate position of being struck down by an outbreak of a particular kind of STEC. It was a serovar known as E. coli 0104H4. There were over 5,000 cases and over 50 deaths associated with this outbreak. It was linked to sprouting seeds. There were some false starts. At one stage it was thought to be cucumbers, but turned out to be sprouting fenugreek seeds. Some kind of wits have called this the sprout break as a result of that. There was a high risk of hemolytic uremic syndrome, and you can see it particularly focused on northern Germany, although it was scattered across the whole of Germany. Females seem to be particularly at risk both of infection and also of the nasty effects of the hemolytic uremic syndrome. There's never really been a satisfactory explanation for that. Perhaps a slightly spurious one is that maybe real men don't eat sprouts, bean sprouts, but we don't really know. This shows you the epidemic curve for the outbreak, and again it highlights that point that you've got skew here, more females than males, both in the number of cases of gastroenteritis and in those that had hemolytic uremic syndrome. You can see that the outbreak was at its peak around the third week in May, came up from nowhere, and then massive amount, and then sputtered away to nothing. And this is a typical kind of outbreaks curve that you'd see. Now, right at the heart of this outbreak was this guy here, Herr Dr. Holger Roeder, who worked at the University Clinic in Hamburg, Eppendorf. So Eppendorf's a suburb of Hamburg. So here he was in one of Germany's largest cities, right at the heart of this outbreak. The hospital was having great difficulty coping. They didn't have enough beds, they didn't have enough bedpans, just coping with that huge number of patients. They didn't have enough intensive care beds, they didn't have enough dialysis capability. But this guy is actually interested in molecular biology, molecular bacteriology, and he thought, well, actually, what about the sequence of the genome of this outbreak strain? Could that tell us what this is all about? But he didn't have the wherewithal to sequence genomes, and so he called on the international community, called international rescue. Now, those of my generation will recognise that that means Thunderbirds arrive and basically save the day. Now, it wasn't quite that dramatic, but the international community did intervene. What he did was he actually got DNA from the outbreak strain, and he sent it to the Chinese Genomics Institute, so called BGI in Shenzhen, just over the way from the border from Hong Kong. They sent it by regular air freight, not Thunderbird 2, but very quickly the DNA was there. It got sequenced very quickly as well. There was a new instrument they'd just taken receipt of that allowed very quick run times called an ion torrent, so-called benchtop sequencing instrument. So they sequenced it very quickly. And then they did something quite remarkable, which was they released the data, the sequence data, into the public domain very quickly as well. I'll leave that bit here. So we were interested in this because I'd actually won an ion torrent in a competition a short while earlier. And within my group, we were kind of gearing up to analyse data from that. So the fact that this iron torrent data suddenly appeared on the internet was actually something very fortuitous from our point of view, that we could actually seize hold of that. And in fact, this guy here, Nick Lohman, who works in my group, happened to note that the data had been released, and he happened to be at, in fact, a bioinformatics and public health conference near Cambridge at the time. And he blogs and he tweets a lot, and he just said to all his fellow bioinformaticians, look, this data's just been released. He had just analysed it, and in fact assembled the data into genome assembly, and that's something he was poised to do because he'd been looking at iron torrent data quite a lot before then. And he put his assembly back into the public domain and said, hi guys, come and have a look at this and see what else you can make of it, what you can tell us about this. So he encouraged this kind of crowdsourced analysis. And something remarkable, similar to what you saw with flu there, within 24 hours of its release, the genome had been assembled by Nick. Within 2 days, it had been assigned to an existing lineage. So it was clear that it wasn't something completely new, like the Andromeda strain coming from outer space. In fact, it did resemble isolates that had been circulating in Germany and elsewhere in the years before. In fact, that was discovered by a guy called Mike, the mad biologist. At least that's what he blogs under that name. His real name's Mike Feldgarten, but he actually managed to get into the data and work that out. 5 days, we had a strain-specific diagnostic test, and within a week we had 24 reports on this open-source wiki. Here's Mike Feldgarten's point about it's not a new strain. And here is just a screen dump. Doesn't come out very well here, but it just shows you all these dozens and dozens of entries on this wiki with people making different comments about the genome. And one of the notable points there is that this was really an international effort. We had, you've got their Spanish names, Polish names, Muslim names, you've got German names, Chinese names. You've got this work going on in multiple continents, in Australia, Europe, America, Asia, all as an international collaborative effort. And in fact, this caught the attention of the science news media. Science and Nature both covered it in their blogs and in their magazines. What were the takeaway messages from this? Well, first, the key point is that pathogens don't bother with passports. This was not a new strain, something that had been seen in Germany similar, but also in Korea, the other side of the world. And when people looked at the genome sequences, there's a guy, Dave Studholm, actually compared the genome sequence with all the known genome sequences from E. coli. He found the closest genome sequence strain had actually come from a patient with HIV in the Central African Republic in the late 1990s. So clearly moving around the world relatively easily. The other interesting thing was that this German S-Tech came from a lineage that circulated in human populations, so-called enteroaggregative E. coli, not from the traditional enterohemorrhagic E. coli, like E. coli 157, which, if you see that organism, you say, that's come from cows, and somehow cow pats have got into the human food chain. But this was a different situation. Clear that bacteria are evolving very quickly, virulence factors jumping around, antibiotic resistance factors jumping around. And it also reminds us that infection hasn't gone away. We kind of think, well, we should all be worrying about diabetes and cancer and heart disease and infections. Well, here is an example where even in one of the most advanced societies on the planet, infection reared its head and actually decimated the society. Now, we use the term open source genomics for what we did, this confluence of high throughput genomics, crowd-sourced analyses, and liberal approach to data release. We were obviously taking a lead from what Jen had already said about flu. It's clear that blogging and Twitter actually made a difference here to things. I mean, my initial response when Nick told me about it was that this is all just froth, and we're here to write papers, and we're going to get judged in the Research Excellence Framework, the REF, next year, and only serious papers in serious journals matter. He kind of said, don't be so grumpy about this. And we managed to square the circle and actually get a paper out of this in the end in the New England Journal of Medicine, which is one of the most highly cited journals. There's a question of whether we've changed the nature of science in this. I think it's clear that drawing on this and on the flu, that this is the way you should handle public health emergencies. And everything should be in the open and quick, and usual rules should be suspended. I suspect ordinary science isn't going to be like this. There is also this issue about, should you do everything through the proper channels of scientific discourse, published journals and so forth, or should you let it all hang out on blogs? Just say what you feel quickly. And there are arguments for and against either stance there. The New England Journal actually published our work, even though they have a policy of saying they don't publish stuff that's already been published before. But it had already been published on all those blogs and stuff, so it was interesting. And it's really exciting that we have this genome sequencing now, bringing the advantages of open-endedness, universal applicability, ultimate in resolution, and with these benchtop sequencing platforms, we can generate data very quickly and cheaply. So, as I say, this culminated for us in a paper in New England Journal. We were very pleased about that. And here we are celebrating with our German collaborators at the top there. And then it turned out that actually another group were working on high-throughput sequencing as well of the outbreak strain. And there's Dave Rasko there in the middle, who was the first author of that paper that came out back-to-back with ours in the New England Journal. In fact, there were 4 groups, all trying to sequence the genome in parallel quickly. It was quite lively competition. We didn't actually win. We weren't the first to get it into publication in a peer-reviewed sense, but we got it into the highest impact journal of any of them, so we're quite happy. So I've added to the handout, there are Jennifer Gardy's slides added on. They've been shared, and